Hello and welcome. In this video, I'll introduce DeepMainmast, a cutting-edge tool for de novo protein structure modeling from Cryo-EM maps with resolutions up to 5 angstroms. Using deep learning and integrating AlphaFold 2 and 3, DeepMainmast provides highly accurate structural models even for maps that are challenging to manually model. DeepMainmast automatically models protein structures from Cryo-EM maps through a series of steps. First, it detects amino acid and atom types using EMAP2SF, a deep learning tool that identifies key atoms like C-alpha, carbon, nitrogen, as well as amino acid types within the map. Next, it traces the C-alpha backbone using a specialized algorithm based on the vehicle routing problem, or VRP. This optimizes how the detected C-alpha atoms are connected to form the backbone, allowing the tool to trace even multi-chain complexes accurately. Once the C-alpha path is traced, DeepMainmast aligns it with your protein sequence using the DAQ score, a metric that evaluates how well the model fits with the map. If you provide an AlphaFold 2 or AlphaFold 3 model, DeepMainmast incorporates it to improve regions with low map density, increasing overall model accuracy. Finally, the tool constructs a complete atomic model using Pulchra and Rosetta CM to refine the structure. These models are scored using both DAQ and DOT scores, which measure local quality and fit to the Cryo-EM map. Now let's walk through how to use DeepMainmast on the Kihara Lab EM server. To begin, you'll need to prepare your files. You'll need your Cryo-EM map of resolution up to 5 angstrom in either MRC or map format. In this example, we're going to model the structure of F420 reducing hydrogenase using DeepMainmast. This is EMD2513 and has a resolution of 3.36 angstroms. You will also need a FASTA file containing the amino acid sequence of your protein, and optionally you can upload an AlphaFold model in PDB format for better accuracy. To submit your job, head over to the Kihara Lab EM server at em.kiharalab.org. Scroll down to the Roadmap section and select DeepMainmast. Now this is where you will upload your files. For the Cryo-EM map, just upload the file in this field. Over here, you can set the contour level for your map. While our tool still works if you leave this blank, it's going to take longer because it will examine all positions, even the noisy parts in the map. By setting the contour level correctly, you will narrow the search space, which allows for faster results. To actually set the contour level for my map, I'm going to use the Visualize with Set Contour Level button to show the map at some contour level. Then I'm going to try out a few values until I can see the protein clearly without noise. That's not good. Could be better. Okay, so it looks like 0.01 is pretty good for my map. And optionally, you can also use Coot or Chimera X to visualize and set the contour level. Next, over here, upload your FASTA file with your amino acid sequences. Now, if your protein contains multiple identical chains, make sure to repeat the sequence for each chain in the FASTA file. So my protein is 3 unique chains, so I have 3 sequences in my FASTA file. But for example, if my protein had 3 copies of this chain, then I'd paste this or copy this 3 times and name them differently. So DeepMainmast can automatically assign the chain IDs properly. If you're using an AlphaFold model, please upload the PDB or CIF file here. But do note that each single chain structure must be in one PDB file and they should be separated by this TER tag. Now, including AlphaFold predictions is optional, but we do highly recommend it for better accuracy, particularly in regions with poor density. If your protein has multiple identical chains, confirm this by selecting yes for the HOMO option. This is important for correct chain assignment for identical chains. By default, DeepMainmast produces a C-alpha only model, but you also have the option to build a full atom model. If you want this, set the Rosetta option to yes. This will make the tool construct a complete atomic model using Pultra and Rosetta CM to refine the structure. However, keep in mind that it is computationally expensive and it will take longer to complete. It usually takes around 5 hours per 1,000 residues. Once you click the upload button and submit your job, DeepMainmast will begin processing it, and you'll see a job ID on the page. Make sure to save the job ID or bookmark the page, especially if you're not logged in, to view the results once the job is complete. If you're logged in, your job will be saved in the job manager for easy access. After the job is completed, head to the job manager and click My Jobs. Over here, you will find all of your jobs. Open up the one that's complete. When the job is finished, you'll get several output models, which you can download. Model 1 is C-alpha only, colored by the DAQ score to show how well the structure fits with the cryo-EM map. Model 2 is similar to Model 1, but it excludes amino acids with low DAQ scores for better reliability. Similarly, if you selected the Rosetta option, Models 3 and 4 will be full atom models, and Model 4 will have regions of low confidence labeled or excluded. All of these models are provided in PDB format, ready for visualization in programs like PyMol or Chimera X. All you have to do is click Download Outputs and you can view them in PyMol or Chimera. Thank you for watching. If you have any questions, feel free to check out the tutorial or reach out for support. For further, more detailed information, please look at our paper published in Nature Methods.